which may be unrelated to any other day of the week as well. How you guys doing? Are you ready for weekend? Yes, 1 week. That first week, we've almost done. Almost got 1/10 of the way through. That's cool. I thought we would start with a song today. Okay? The song today is the first one that you will hear that's not written by me. It was actually written by one of my students back when she was a TA of mine a few years ago, and she got into the songwriting thing. I think she did quite a good job. It's called the Citrate Sonata, and it's about the citric acid cycle. It's to the tune of God Rest Ye Merry Gentlemen, and I hope you will join me. Our fats and carbs get broken down to acetyl-CoA. Oxaloacetate combines in cycles TCA. The product of reaction 1, citrate is its name. Isocitrate, the product that ensues. Atoms got moved. Isocitrate is the product of step 2. An oxidation soon occurs, reducing NAD, and alpha-ketoglutarate resulting from step 3. From here we could make glutamate, that is if there's a need. Don't forget that we lost a CO2. Yes, it is true. In reaction 3 we lost a CO2. So what's the point of all these steps? Well, let me tell you, friend. We use electron carriers in working towards our end of synthesizing ATP, a metabolic trend. Oxidize and then oxidize some more. Here in step 4, ketoglutarate gets oxidized some more. The enzyme with cofactors 5, including TPP, lipoate, FAD-CoA, and also NAD. A succinyl that's on CoA is what gets made, you see. This reaction occurs so favorably. Don't you agree? It's a good reaction energetically. With 4 more steps, we're halfway there, so let me summarize. When CoA is lost, we see that GTP is synthesized. The succinate that is produced will soon get oxidized. FAD goes to FADH2. What did we do? We made fumarate and FADH2. Add water across the double bond and malate we create. With one last NAD we can then dehydrogenate to give a final product of oxaloacetate. It's removed and this lowers delta G. Oh yes, indeed. It's through pulling that this last step can proceed. Very good. So, all right. That's, yeah. Okay, so that was, I just said that wasn't even one of my songs. That's one of my students' songs. So, all right. Now, if you like the metabolic melodies and you would like to participate in a photo shoot today where they're going to be taking some pictures of me with students looking like they're singing metabolic melodies, you don't have to actually sing. Okay. If you're free at 3:30 this afternoon and would like to participate, I need 2 or 3 students more, maybe even 3 or 4 students more. If you're interested, see me after class and I'll tell you where it's all at and everything. You won't have to sing. You just have to look and pretend like you're singing because they're going to take a picture. Okay. I promise I won't embarrass you. All right. So we've done the citric acid cycle. We've done the glyoxylate cycle. And I said I was going to tell you a little bit about the glyoxylate cycle. And I think what I will do with that, it's probably easier if I write it down for you. So I'm going to write you some notes about the glyoxylate cycle just to answer some common questions. I'll just tell you what some of them are right now. So some of the common questions. Well, when does the cell know to do the glyoxylate cycle and when does it know to do the citric acid cycle? Because they're both present, right? They're both going on. And the answer is the same to this as it is to all. This thing cuts in and out. This to all of the metabolic pathways in the cell. It depends. And it depends on the cellular needs. Okay? Since this glyoxylate pathway produces an extra oxaloacetate every time it goes around, since that happens, that oxaloacetate can be used for things. The cell needs to make some aspartic acid. Guess which pathway it's going to run? That one. Glyoxylate. If the cell needs some glucose, which pathway is it going to run? Glyoxylate, okay? If it needs, on the other hand, energy, man, we got to get some out of here, we got to get some ATP, it's not going to do the glyoxylate cycle. Because I didn't show you in going around, but I'll tell you here, since we bypass those two decarboxylations, we lose two NADHs. So the glyoxylate cycle doesn't generate nearly as much energy as the citric acid cycle does. So if the cell needs energy, it'll go the citric acid route. If it needs other things like glucose or aspartic acid or something, it's going to go the glyoxylate route. We don't have that option. As I said, we don't have the option of making glucose from acetyl-CoA because we cannot run the glyoxylate cycle because we're missing those two enzymes, isocitrate lyase and malate synthase. We don't have those. Bacteria, yeast, and plants all have them. Okay, so, and as I said, I'll write up a little blurb for you for that on the highlights for next time. What I want to do now is turn our attention to lipids and cell membranes. And it's appropriate that we talk about that at this time because, as we're going to see, we're soon going to be talking about electron transport and oxidative phosphorylation, two very important processes that occur in the mitochondria, and we're going to discover they occur in the membranes of the mitochondria. So it's important, therefore, that we understand something about the composition of the membranes, and then we can talk about the actual processes that are occurring in them. We've said a little bit about this before, so some of this will be review. You remember from your cell biology class, and probably also from things we talked about last term, that the membrane of all cells is composed of a lipid bilayer. The lipid bilayer schematically is shown sort of like this, and there's a variety of schematic drawings that are out there. This schematic drawing, however, is 100% backwards, right? Does anybody see anything stupid in this diagram? It's totally hydrophobic. They've got the thing of the hydrophobic tail sticking out, and they've got the hydrophilic thing sticking in the middle. Okay? Now, why in the world? This book has already gotten several things wrong. It's not off to a good start this term. Hopefully you don't have the same start that you're off to this term. Okay? Not a good idea. Don't want to have that happen. I don't want you to get screwed up like this. Okay? So, the real lipid bilayer has these red guys on the outside and all the green guys on the inside. So red out, red to the bottom, red to the top, and green in the middle. That is the composition of a lipid bilayer. I'll remind you, these green guys are long-chain fatty acids, and those long-chain fatty acids are very, very nonpolar. The lipid bilayer, therefore, if we were to go from top to bottom, goes polar, nonpolar, polar. That actually provides a pretty good barrier for things that want to cross. If something's polar, it doesn't get past the first level. If something's nonpolar, it doesn't get past the second level. Okay? So polar, nonpolar, polar provides a pretty good barrier for cell. It's not an absolute barrier. Okay? There are ways for things to get in, but it's a pretty darn good barrier. Things don't cross that membrane very easily by themselves. There are 4 exceptions to that. There are 4 things that cross that lipid bilayer easily. One of them is water. One of them is oxygen. If oxygen didn't cross that layer easily, we'd be in trouble. We rely on oxygen. Carbon dioxide crosses that layer very easily. That's also important because we produce carbon dioxide. We want to get that out of the cell. And the 4th, unfortunately, is carbon monoxide. It crosses that lipid bilayer very easily. Now, these 4 things don't require transport systems to move across. Transport systems, you may recall, involve proteins that grab something specifically and transport it across the membrane. These 4 guys don't require that. Okay, well, let's look at fatty acids. You've seen fatty acids in organic chemistry. I'm sure you've seen it in a variety of places, and this will be a review for you. But fatty acids come in 2 general types. One type, of course, being the saturated fatty acids, shown on the top, as drawn as a relatively straight chain. And the bottom, which is an unsaturated fatty acid known as oleic acid or oleate, which has a bend. Okay? Now, when you look at this guy on the top, you think, okay, that's mostly a straight chain. This guy down here is mostly a bend. That's a little inaccurate because this guy up here can do all kinds of rotations and so on and so forth. But suffice it to say that when it's in its own native environment surrounded by other fatty acids, it pretty much does flatten out like that, even though it's got a lot of ability to do rotations and so forth. Okay? Whereas this guy, this bend, really is an absolute thing. It cannot be retwisted or something like that. The bends that we see in the unsaturated fatty acids, of course, arise from double bonds. And the double bonds that we have in unsaturated fatty acids that are produced biologically are almost always cis. They're almost always cis. We'll talk about trans fats later, and trans fats arise not as a result of biological processes for the most part, but rather by the chemical treatment of food. Okay? So, naturally produced biological fatty acids are almost always in the cis configuration. Okay? Now, a little nomenclature. When we talk about fatty acids, one of the things that you hear about is omega-3 fatty acids, right? Everybody's heard of omega-3 fatty acids, right? And omega-3 fatty acids are supposed to be good for you, right? Well, it might be useful to know what omega-3 fatty acids are. I bring this up because there's two different nomenclatures that are used to describe the position of double bonds in an unsaturated fatty acid. I want you to be aware of both of them. Okay? So the omega system is one of them, and that's the one we'll do first. What the omega and the 3 is referring to is the position of the double bond that's closest to the methyl group at the end. So if we talk about something that's omega-3, for example, we would be talking about a fatty acid that has a double bond 3 carbons away from the end, the methyl end. Okay? There's 2 ends to a fatty acid. There's a methyl end, there's an acid end. Okay? So the omega system counts from the methyl end. It counts to the very first one it gets to, and if we had a double bond right here, that would be an omega-3 fatty acid. Everybody with me? Now, the other system that's used to describe the double bonds in fatty acids describes all of the bonds in the fatty acid, and it starts counting at the other end. Okay? So the other system says this is carbon number 1, okay? And it counts in that direction. All right? So carbon number 1, actually this is showing, it says omega, that's not right. That's actually the delta system. The other system is called the delta system, and it numbers starting right here. Okay? So if we start with the carboxyls number 1 and we count over, we get to the first fatty acid that becomes delta, let's say delta 9. That's a very common one that we see. Delta 9. It's 9 carbons away from the first carboxyl group. Okay? With the delta system, we number all the fatty acids. We call them delta 9, delta 12, delta 15, for example. That's a fatty acid that has 3 double bonds in it. One at position 9, one at position 12, one at position 15. Okay? Now, the delta system is what biochemists tend to use more often. So we're probably going to talk more about delta than we are about omega in here. Okay? The delta system is how we orient things, and one of the ways I'll orient you is to tell you that if we look in the delta system at human beings cannot make double bonds anything past delta 9. So delta 9 they can do, but they can't go anything past delta 9. So, for example, if we talk about an essential amino acid like linolenic acid, it's delta 9, 12, 15. Okay? A 9, 12, 15, we can't make that. We can make a 9. 9 is oleic acid that you saw before. In fact, I'll show you oleic acid with that numbering again. Okay? If we look at oleic acid, here's oleic acid. 1, 2, 3, 4, 5, 6, 7, 8, 9. So we can make oleic acid. 10, 11, 12, couldn't make that one. 13, 14, 15, couldn't make that one. That's why we refer to things that have double bonds further out in that chain as being essential. If they're not in our diet, we're host. We have to have them. So we need to have unsaturated fatty acids, and we need to have unsaturated fatty acids besides oleic acid, which is this one that we have here. Everybody with me? So keep the distinction between the carboxyl, number 1 for the deltas, and the methyl as number 1 for the omegas. Okay. Okay. And there is another designation. In fact, this is an accurate designation. Now they're properly showing you the omega-3, 1, 2, 3. There's the double bond. And you can see an omega-3 fatty acid as a result of that. Okay. There's a list here. My aim in showing you the list is not to get you to memorize a bunch of stuff on the list, but to make you aware of. I think you should certainly know the names of some of the common fatty acids. You should know what palmitate is. We'll talk a lot about palmitate this term. Palmitate is a 16-carbon saturated fatty acid. Yes, I think you should know that. Okay? Stearate, 18-carbon saturated fatty acids. It derives its name from steers, where we get saturated fatty acids. Okay? Oleate, 18 carbons, 1 double bond, position 9. Okay? Linoleate, 18, 2 double bonds. Linolenate or linolenic, same thing, 3 double bonds. All right? The last one here is interesting. It's called arachidonate, and we'll see arachidonate is a precursor of the class of molecules we refer to as prostaglandins. Okay? Arachidonate has 20 carbons, and it has 4 double bonds. I'm not as concerned that you understand, that you memorize where the bonds are. I think for olate, you should know its position 9, but I don't care about these other ones down here. But I do think you should know linolate has 2, linolenate has 3, and arachidonate has 4. These are important fatty acids, and they're essential. At least these two guys here are essential. This guy we'll talk about later. There's a very brief thing in today's lecture, but it won't take me just a minute. But we'll talk later about the synthesis of prostaglandins, which are a fascinating group of molecules. Okay. Now, if we look, and I think we saw this last term, if we look at the generalized structure of a molecule that we might find in a lipid bilayer, it has a structure that looks like this. Okay, we've got a glycerol backbone. These would be the glycerophospholipids. And we have sphingolipids also, and sphingolipids don't have glycerol in them. We have glycerol backbone, 2 fatty acids. We have a phosphate, and that phosphate is commonly linked to something else. Okay? So if we look at just this portion, we call this the phosphatidyl portion. And whatever is out here is the rest of the name. So if this were, let's say, ethanolamine, we would say this is phosphatidyl-ethanolamine in our nomenclature. Okay. Now, this guy, as you see, has a polar head, and it has a non-polar tail. Things are going to arrange themselves in that lipid bilayer accordingly. There's a much worse representation of the same thing, but you can see there's the glycerol backbone in black, fatty acid number 1, fatty acid number 2, and here's the other stuff hanging out here. This is called phosphatidic acid because nothing has been attached to the phosphate yet. So phosphatidic acid is a glycerophospholipid as well. If we attach something here, it becomes a phosphatidyl something else. Now, these R1 and R2 refer to different fatty acids that are up here. Fatty acid number 1 is usually saturated, and fatty acid number 2 is usually unsaturated, although those are general rules, not absolute rules. There are some of the things that can be added on to that phosphate group. So we saw this last term also, phosphatidylserine, phosphatidylethanolamine, phosphatidylcholine, phosphatidylglycerol, etc. A variety of things that can be attached to that phosphate group at the bottom there on position 3. So these are all things that we might find as part of the glycerophospholipids scheme. They would be all things found in phosphatidylglycerol compounds. And there's some structures. Again, we're not going to memorize structures, but you can see how they fit in. There's the glycerol backbone, phosphate, in this case serine attached, choline, et cetera, et cetera, et cetera. All right, the other class of, a very common class, I shouldn't say the only class, but the other common class we see of lipids in the lipid bilayer are those of the sphingolipids. And sphingolipids derive their name from the fact that they are related to the compound known as sphingosine. We will see later in the term that the sphingolipids are made using serine and palmitic acid, and we'll see that later. Sphingomyelin is a very interesting sphingolipid. It is a sphingolipid that is very common. In fact, it's very, very abundant in the myelin sheath of nerve cells. So sphingomyelin is very abundant in the myelin sheath of nerve cells, and it has a sphingo structure, which I'm not going to go through. We did that some before, so I'm not going to go through all that again. But at the other end, you see here is a phosphate that's been linked to a choline out here. So this gives sphingomyelin its structure. Sphingomyelin is one of the few sphingolipids that we will talk about that has a phosphate in it. Most of the sphingolipids don't have phosphate in it. In fact, they have something else usually attached in the place of the phosphate. And I'll show you that in just a second. So a cerebroside, for example, instead of a phosphate, has a simple sugar attached to it, in this case either glucose or galactose. We commonly do find sugars attached to the sphingolipids. So if we have a simple sugar on there, it's called a cerebroside. If we have a complex sugar on there, it's called a ganglioside. Okay. Cholesterol, we mentioned last term also, is also found in the lipid bilayer. Cholesterol is an important component of membranes. And cholesterol affects the chemical properties of those membranes. Okay. And cholesterol, as you can see, is a mostly nonpolar molecule. This OH group is the only polar portion of cholesterol that there is. So most of cholesterol remains embedded in the nonpolar portion of the lipid bilayer. Okay. Now, there are cells on Earth that have to live in some pretty bizarre environments. And virtually everywhere we look on Earth, we see growing living cells, which is pretty remarkable. And when people have characterized the organisms that live in some of these bizarre environments, they discovered a whole new class of organisms called Archaeans, Archeons. And the Archaeans, because they have to live in unusual environments, not surprisingly, in many cases have different lipid bilayer structures than do cells that don't live in those environments. Okay? So this is a very caustic environment. You don't want your lipid membranes falling apart chemically in that environment. And so when we look at the Archaeans and other things that live in those unusual environments, we see a difference. And one of the differences we see is shown right up here. Now you might say, well, it looks very much like the other one before, except for you just turned it upside down. And why they drew this upside down, I don't know why. But there's another important difference. In the case of the glycerophospholipids I showed you before, they had an ester bond. That was a carbon with a double bonded oxygen there and a carbon with a double bonded oxygen there. These are known as ether lipids, ether glycerolipids, because they have an ether bond. COC is an ether bond. And this ether bond appears to give these guys greater stability in the environments in which these cells live. So again, we see life adapting to the environment in which it finds itself, and that adaptation occurs all the way down to the chemical level, in this case, as we see here. Now, I like these figures a little better than those other ones because I think they give you a little bit better feel for the similarities. There's a sphingolipid, there's a glycerophospholipid, and they're really not, from an appearance point of view, very different from each other. Here's an archaeal lipid, which has the ether linkage. And again, we don't really see big differences in the way they appear. We see bigger differences that occur as a result of double bonds than we see of the different classes of lipids. So let me pull up the double bond for you here. Yep. So if we had a lipid bilayer of solely of saturated fatty acids in the glycerophospholipids, we would see that. On the other hand, if we have double bonds, it's much more chaotic. Now, I hope you remember that when we put more double bonds into a fatty acid, we lower its melting point, we lower its membrane fluidity. And the reason that that melting point lowers is this chaos that you see right here. Okay? This chaos right here. It doesn't take much energy to get these guys moving around. So the more we unsaturate the fatty acids in the glycerophospholipids and also in the sphingolipids, the more we unsaturate those fatty acids, the lower the melting point, the more chaotic that environment of the lipid bilayer actually is. Let's see. I'm kind of moving kind of fast, aren't I? Nobody says anything.
So if we eat more Arachidonic, does that mean we make more prostaglandins?
I'm sorry, say it again? If you eat more arachidonic, does that mean you make more prostaglandins? The answer is no, and you probably wouldn't want to do that. Okay? Prostaglandins are made at the site of the cell membrane, as we'll see in just a minute, and they're made and they're broken down very, very quickly. So prostaglandins do some things that you would consider good. I hate using that term, and some things you would consider bad. Some of the good things that they're involved in include processes in the body, including some short-range signaling processes that are very important. Some of the bad things include pain and certain types of muscular contraction that you don't want to have. So it's a mixed bag. But in general, no. And in fact, you have a hard time getting much because arachidonic is broken down pretty readily. Okay. Well, let's see. Let me slow down. Somebody tell me a good joke. You guys want a good joke? Okay. So you got two grizzly bears. Okay? You got one up in far northern Alaska, okay, where it is colder than you can imagine, right? And you got one in the tropics of Montana. Okay? And they both go at the same time, and they both go for a swim in the water. Okay? You got to bear with me on this one. This is a little stretch, okay? You heard it already? Oh, I'm not there yet. I like him. Okay? They both go for a swim. Which one dissolves first? The one in Alaska. It's the more polar one. Okay, who told me that joke? Blame him. No, I love that joke. It's very cute. Very cool. Okay. Polar, you see it fits. This is relevant. This is not just telling a joke. It's relevant for what we're talking about. All right. I actually knew a guy one time, and his name was Ion. Is that a weird name for a kid or what? Ion. You know? His parents would keep an eye on him. I guess that's probably where they had... No, I'm not making that. That's not the joke. I mean, that was actually really his name. His name was Ion. All right. All right. So let's think about lipid bilayers. How do we get lipid bilayers? If we take... you guys know the old thing from organic chemistry, how a soap works. How does a soap work? Well, you take molecules that have a long tail and a polar head, and you mix them together, and what happens is soap works by the fact that you have grease on your hands. Grease is nonpolar. The soap molecules arrange themselves in a micellar type structure that you see here, and the grease associates in here, and a little droplet gets carried away because these guys associate with water. They actually drew it in the right orientation here, right? So that's how soap works. That's not how a membrane works, and that's not how a membrane forms. Why doesn't this associate it like in a lipid bilayer instead of in a circular structure? The answer is because soap is made with only a single tail. Taking polar head and sticking two tails on it actually keeps this from forming. They squeeze each other out, and instead of going circular, they go linear. So when that happens, they go to this. These guys can no longer arrange themselves in that spherical structure that we saw before. They arrange themselves into a lipid bilayer. And that formation is actually built into the chemistry of these molecules. They will, all by themselves, assume a lipid bilayer if you mix them together. Now, that's a very cool thing because it means that you can make membrane-enclosed structures yourself in the laboratory very, very easily. All you have to have are some glycerophospholipids, some sphingolipids, and you take and you put them into a sonicator in a solution of water, and they will assort themselves and arrange themselves around structures. They'll make little cell-like structures is what they will do. Usually they're pretty small, but they'll make little cell-like structures. That turns out to be a very, very useful thing, okay? Because here's how we would do. Here's what they look like. Here's that green nonpolar portion. Here is the one polar side. Here's the other polar side. So this kind of looks like a cell. Now, why would we want to make one of these in the first place? Are we creating artificial life? Well, by creating artificial membranes, I hate to tell you that creating life is a lot more complicated than that. But instead, we create structures like this in the laboratory to do something very, very important. We make structures like this to put something on the inside that we want to introduce into a cell. Okay? Now, let's say I've got a molecule, I've got the world's greatest drug that I've just created, and the thing is just polar as all get out. And I want to get it into cells, but it won't go across the cell membrane because it hits that polar stuff fine, it hits the nonpolar portion, and it doesn't make it into the cell. However, if I take my drug, and I'll show you that now. If I take my drug and I mix it in a flask, like you see here, here's my drug in red. Here's the lipid bilayer that's in the bottom. I shake it up, I sonicate it. What happens is those guys start forming those artificial structures, and look, they start enclosing the drug inside of them. Because on the inside, that's where the water is going to be. Now, when I take this guy right here, which has a membrane almost exactly like a cell, and I scrunch it together with a cell, guess what happens? The membranes fuse. And now whatever is in here gets introduced into the inside of the cell. It's a very good way to introduce polar molecules into cells. Very, very cool way to do that. Okay? And it doesn't take that much effort. They're actually fairly easy to make. These guys have a name. They're called liposomes, Liposomes. They're a man-made structure that is very much like a cellular plasma membrane, and they contain goodies on the inside. Everybody got that? Yes, Virginia.
Is this ever used by either bacterial viruses to also enter cells as we can do it with?
Oh, that's a good question, actually. Do bacteria or viruses do this to get into cells? They actually have other tricks that they use. So bacteria are a little bit different. Viruses have specific structures that bind to specific membrane structures on cells, and they have a little injection port where they inject their nucleic acids. They don't do that, although one could imagine that one could actually infect cells if you had bacteria in this flask, right? Because if you have bacteria in here, now you fuse it with a cell, you just introduce something into the cell that's probably not going to play good with the interior of the cell. But yeah, interesting question. Yes, Laurie. They will fuse if they're next to each other. They will. And so it means you've also got to be careful with this because there's how are you going to control where it's going to go, where you want it to go. And so while it's useful, a very useful tool in the laboratory, in the real world, you have to some other considerations for it. But yes, you can. The association is simply enough to do it. Yes?
Can you ever stick targeting proteins into the cell membrane?
Can you do what?
Can you ever stick targeting proteins into
You mean like right here? You mean sticking it right in this thing?
Yeah.
That's a good question. I never thought about that, but I don't see any reason why you wouldn't be able to do that. The problem that you would run into would be getting the protein from here into here. And you say, well, that's not a problem. And in fact, it is. Because if it's a membrane protein, it's going to be mostly nonpolar. It's probably not going to be very soluble in this mess up here. But I don't see any reason in practice, in theory, why you couldn't do that if you could somehow keep it solubilized. Okay? There's a new technique. There's a patent right there. Okay. Let's see. Where am I at here? We're not going to do measurements. We're not going to worry about that. Membrane proteins. Centrifuges are really good. Okay? With a centrifuge, you can take a bunch of cells, you can bust the cells open, split them open, that leaves membranes and all the guts of the cell that come out. The guts of the cell that come out are pretty much liquid. The membrane itself, of course, is not liquid, it's a solid. And so you can separate the liquid from the solid by centrifugation. It's pretty easy to purify membranes. And when you purify membranes and then you take the contents of those membranes and you run them on a SDS polyacrylamide gel electrophoresis system, you discover that those membranes are full of protein. Okay? We talked about membrane proteins a little bit last term, and so this is just a documentation of that. What we see looking at different membranes is their composition of protein varies tremendously. One of the places where we see the absolute most protein in membranes is in the mitochondria. And that's partly because the mitochondria are doing so many things in their membranes. They are loaded with proteins. Okay. Now, here is a schematic representation of a membrane with some proteins in it. And I have a little different way of naming the proteins than your book does, and so I'll remind you of what my designation is. A is a protein that is what your book and I agree on. That one is called an integral membrane protein. Integral means that it has ends that project through both sides of the lipid bilayer. Okay? E and D, so we can see here we have integral, integral, integral. Your book refers to E and D as peripheral membrane proteins. I give a more specific name than that. Okay? I call these guys associated membrane proteins. They're associated with something that actually is in the membrane, but they themselves are not in the membrane. They're associated with a membrane, but they're not in it. There's a third group of proteins, not shown on here unfortunately, where the protein doesn't go through both layers of the lipid bilayer. It only goes through one. So we can imagine it would sort of fit like here. Okay? It's partly in the membrane. It's not associated. All right? That is what I refer to as a peripheral membrane protein. Because it's peripheral, it can easily be removed and taken out of the membrane. So peripheral membrane proteins project through one layer of the lipid bilayer, not both. But they're in the membrane. They are in the membrane. Associated proteins are associated with something in the membrane, but they themselves are not in it. And there's a fourth term that I use to describe membrane proteins. And that one is called anchored membrane proteins. Now, an anchored membrane protein is covalently attached to something that is stuck in the lipid bilayer. And I'll show you an example of that in a minute. It's covalently attached to something that is stuck in the lipid bilayer. So an anchored membrane protein is covalently attached to something that's stuck in the lipid bilayer. So 4 different classes of proteins for our purposes. Now I'm going to talk about a real cool protein. I've got $1 million idea for somebody, maybe $1 billion idea for somebody, and I throw it out to every class I teach. If you guys want to do this, you can get rich, okay? It involves this protein. What does this protein do? All right? You can kind of sit back and relax on this one. I've been going fast enough as it is anyway. This protein is a fascinating bacterial protein called Bacterial Rhodopsin. Okay? How many people have ever heard of Bacterial Rhodopsin? Okay, a little bit. So Bacterial Rhodopsin is a protein that it resides in the membrane of bacteria. It is an integral bacterial protein. And it is a protein that is active in the light. Okay? Active in the light. Now, we see its sort of schematic representation here. And in the middle of this complicated structure, there's a molecule of vitamin A. Now, vitamin A, you remember, has a role in your eyes because it allows you to see. And we'll talk at the end of this term about how vitamin A allows you to see. But suffice it to say that vitamin A is light sensitive. It has double bonds, and those double bonds are isomerized by the light. Vitamin A changes its shape from straight to bent just by being in light, and back to straight. We have a protein in our eyes called opsin. Opsin is vitamin A linked to a membrane protein in our eyes. Okay? Bacterial rhodopsin has vitamin A in it, and light does something with it. So I want you to think about the middle of this thing. This middle of this thing is like a barrel. Maybe a barrel's a little big to think of. Maybe a narrower chamber. Maybe a little tube. Okay? And in the middle of this tube is a molecule of vitamin A. And vitamin A does two things. In the light, it can start out straight. In the light, it bends. In the light, it goes straight. You put it in light, it's going to do doink, 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 doink. Okay? No biggie, right? But mechanically, that's what this guy is doing. Doink, doink, doink. All this, right? And it's going really rapidly. This tube that we find in here is a chamber where protons fit in very nicely. And we're going to talk about electron transport later, and it'll be relevant to this as well, so it's part of the reason I'm introducing it now. Proton gets in here, light hits this thing, it goes doink, and it kicks the proton. Doink, and it kicks the proton. Okay? So it's kicking protons actually outside the cell. Proton comes, it kicks it out. Proton comes, kicks it out. Proton comes, kicks it out. As long as there's light there, it keeps kicking out protons. What happens then to the concentration of protons outside the cell compared to inside the cell? It's going to go up. So we have a voltage across that cell. We've just created potential energy. We've just created potential energy. Now, what we're going to see when we talk about oxidative phosphorylation next week is that our mitochondria are doing something very similar, but they're not using light to do it. They're kicking protons outside the mitochondrion, getting a high concentration of protons out in the cytoplasm, and they want back in the mitochondria. So our cells have a structure called complex 5 that allows the protons to come back in. And when the protons come in, a little turbine turns, I'm not making this up, and ATP is synthesized. I'll show you an animation of this next week. Okay? So high proton concentration turns a turbine, makes ATP. Now, here's the $1 million idea. Bacterial rhodopsin is a protein from bacteria. We can get the coding sequence for it very, very easily. Right? We can take that coding sequence and we can put it into another organism very, very easily, right? How many people in here are into aquariums? Anybody? You like my favorite aquarium thing was the little clear fish, right? You can see right through them, right? You guys have seen these things, haven't you? Well, now what if I took that protein and I put it in the mitochondria of these little clear fish? And they start, when I turn the light on, they start pumping protons out. The protons went back in. What's going to be made? ATP. Do I have to feed the fish? I've just made a photosynthetic fish. Now, see, I just gave the idea away. If anybody goes and makes $1 billion on this, I want a chunk, okay? But seriously, you can do this. I actually talked to somebody who knows something about mitochondria. Okay? And I said, what do you think about this? And he said, it's a very interesting idea. He said, why don't you try it? And I said, well, I've got a few other things to do. So I throw it out. Seriously, go do it. It'd be kind of cool to do. You make yourself a photosynthetic fish. Now, it would not be photosynthetic in the sense that a plant is. Okay? A plant takes light energy and it pulls CO2 out of the atmosphere. It has a lot of structures for doing that. So it wouldn't be photosynthetic in that you would still have to feed the fish a little bit so it had the nutrients that it needed. The plant makes its own nutrients. But the amount you would have to feed this fish is pretty tiny. The limited amount of nutrients that you would give this fish would be more than taken up by the amount of ATP that this thing is making, which I think is pretty cool. Okay, there's my idea. All right. So that's my billion-dollar idea for the day. And it comes using Bacterial Rhodopsin. All right. Porin. We talked about porin last term. I'm only going to show you very briefly what is with porin. Porin is what the rain is doing outside, I think. But porin, you remember, is a membrane protein. We talked about it last term. It had, what was the distinction about it? Why did we talk about it last term? Protein structure. So it flips. It's got the outside's got the nonpolar stuff, so it can interact with this nonpolar lipid bilayer. On the inside, it's got a polar region that can associate with water because porin's job is to let water pass through it, and that's what it does. Okay, that's porin. Now, prostaglandins. Megan asked me about prostaglandins. So, about arachidonic acid. Prostaglandins are made inside of cells, and again, we'll talk about this later in the term, so we'll actually deal with their metabolism later. But schematically, prostaglandins are made by an enzyme called prostaglandin synthase. And there are several of these. And you see that they're associated. This guy is what we would describe as a peripheral membrane protein. Notice that it is projecting through a portion of the lipid bilayer, but not through both parts. This is a peripheral membrane protein. This is the inside of the cell down here. Outside is up. Okay? And what this guy does is it grabs arachidonic acid and it makes prostaglandins. And the way that arachidonic acid is produced in the cell is by cutting a fatty acid off of a membrane component. So if we have arachidonic acid right here and the cell needs to make prostaglandins, it will cut that arachidonic acid off. This guy will grab it and make prostaglandins. So being located in the membrane where the arachidonic acid is being produced is a very, very useful thing for this enzyme. So and the other thing about peripheral membrane proteins is they move through this membrane very fast, very rapidly, back and forth in this direction. Okay, let's see. Oh, one last thing and then we'll finish for the day. And that's the anchors. I said we had anchored proteins. I said an anchored protein was a protein that was covalently attached to something that was stuck in the lipid bilayer. Here are some of the things that can anchor a protein to a lipid bilayer. Okay? So here is S-palmitoylcysteine. You don't even know these names. Don't worry about it. It's the structures that are more important. These molecules that anchor a protein to the membrane all have a long tail like a fatty acid and a portion that is attached to the protein. So you see the attachment to the protein here. You see the long tail that's going to stick into the lipid bilayer. And this is where the anchor is attached. There's the anchor. There's the protein. Clear as mud? I think it's time for weekend. See you guys Monday. If you're interested in the photo thing today, let me know. I need 3 or 4 people. How you doing?
They're good.
Good?
What do you think about HHMI for what you just said? Is that possible?
Oh, it's a bigger project than that.
Too big?
Yeah, I talk about it simply putting it in, but you've actually got to do some engineering. So
But somebody could do it. It'd be kind of cool. Do it.
Such a good idea.
Yeah, do it.
Do that fish idea for a summer.
That's what somebody just asked me. It turns out it's a lot more complicated than that, because I described that putting it into an organism is pretty simple, and it's actually more complicated. But it's a theoretically possible thing to do.